I also want to point out that on the phone is Dr. Kioka from Harvard University. Also having joined us in the middle of these deliberations is Dr. Susan Ross from University of Pennsylvania and Dr. Lori Zolef of Northwestern University. Well, welcome. And Dr. Ms. Molino, our public member. We're going to move to the next protocol under consideration. And that is protocol number 1188, Phase 1 Gene Transfer Clinical Trial for Spinal Muscular Atrophy Type 1, delivering the survival motor neuron gene by self-complementary AAV9. The PI is Jerry Mendell. He currently serves as the director of the Center for Gene Therapy at the Research Institute at Nationwide Children's Hospital and holds professorships in neurology, pediatrics, pathology, physiology, and cell biology at the Ohio State University. He also directs the neuromuscular research program at Nationwide Children's Hospital. So welcome, Dr. Mendell. And if you could introduce the other members of your team who are here today.
I will. Thank you very much. We appreciate you reviewing our protocol and the considerations on the questions. My team is right here, and on the far right we have Reed Clark, who is our vector manufacturing expert. We have John Kissel, who is an SMA specialist. We have Arthur Burgess, who's a research scientist and an accomplished member of the team developing the SMA animal model that I'll speak about. Then we have Brian Kaspar, who really did all of the preclinical work on this protocol, and another research scientist we had that accompanied Brian in the research work, and that is Kevin Faust. And their names appear here in the introductory slide. So without further comment, let me just say our presentation objectives today will be to talk about the study goals and the rationale for this clinical trial, describe the preclinical studies in preparation for this trial, present the toxicology studies that we have completed and discussed with the FDA and planned in preparation for the clinical trial. And then I'll summarize the clinical protocol that we plan to go forward with. And of course, address any questions that the RAC has. And we did submit the responses to the initial questions. Let me just talk a little bit about the disease. Spinal muscular atrophy refers to a group of disorders that is characterized by a loss of motor neurons that map to chromosome 5q. For this specific disease protocol, we're really talking about one of the subtypes of spinal muscular atrophy, that's SMA type 1. This is the most severe of this is the most common genetic cause of infant mortality. It has an onset before 6 months of age, with signs of weakness by 3 months in virtually all cases. The disease is characterized by severe hypotonia, progressive limb and bulbar muscle weakness, which manifests as a weak cry, a poor suck, and finally respiratory failure, with almost all children with this disease dying by age 2. In terms of the pathogenesis, it's quite clear that SMA is caused by a homozygous mutation of the survival motor neuron gene. And it's important to understand that the survival motor neuron gene that we're talking about is SMN1, and this is a telomeric gene on chromosome 5. But it's very important to understand also that there is a nearly identical copy of this gene that is referred to as SMA2 that's centromeric. And you'll see the importance of this in the discussion here. SMN1 produces the full-length transcript. and this is the gene that has the significant mutation and is lost in the disease. SMN2, which is this nearly identical gene, produces an alternatively spliced message that lacks one specific exon, and we refer to this as SMN delta 7. The importance of this is quite clear, that SMN 7 is unstable and is rapidly degraded, but it can be alternatively spliced and produce low functional levels of the SMN protein. And this is nicely demonstrated on this slide, which is the real understanding of the pathogenesis of this disease. At a pre-mRNA level, we have exon, this shows exon 6, 7, and 8, and in this we have producing when the full message is produced, we have exons, we have all three of these exons represented, and we get full-length normal SMN protein levels. Normally what happens is that we have SMN2, which it has a deletion of exon 7 because there's a C to T mutation here. And so at the transcript level, you see we have just exon 6 and 7 represented, but because there is occasional alternative splicing, we have full-length protein, full-length SMN2, and it actually can produce low SMN levels. In that context, when SMN has a mutation and is not represented, the disease can be modified by how much SMN2 protein is expressed. And you'll see that here in the next slide, that shows the classification of SMA, the disease that we're talking about, actually correlates with the number of SMN copies. There is a very, very severe disease which we think has a prenatal onset, where babies die before 1 month of age, and these infants have only one copy of SMN2, and it's not enough to compensate for the loss of SMN1 in any significant degree. On the other hand, the disease that we're targeting is called SMA type 1. It presents between birth and 6 months with a death before 2 years of age, but has 2 copies of SMN2 that can help the child survive up until this time point. If there are more copies, we see milder disease, that is, more copies of SMN2. For example, if we have 3 to 4 copies, we can have a disease with onset before 18 months, but with infants that survive past 2 years of age. And there are other infants who have onset after 18 months, sometime between 18 months and 3 years of age, who can live into adult life. And they, too, can manifest their disease, which we call SMN type 3, because they have the excess copies of SMN2. And finally, there can actually be an adult onset disease if there are multiple copies of SMN2 that compensate for the loss of SMN1. And here we see 4 to 8 copies would actually produce an adult onset. So we have really a continuous disease where we see compensatory responses depending on SMN2 copies that compensate for the loss of SMN1. Now, it's very important to understand the rationale for the protocol that we have used. is really dependent on what we know about the disease, and that is that there is a very rapid loss of motor neurons in infancy, and we're talking about now SMA type 1. And with elegant studies, for example, by Richard Finkel, comparing CMAP potentials. CMAP potentials are compound muscle action potentials that represent the number of motor neurons. And we can see that within a very early onset after birth, there's a very rapid loss of CMAP potentials that clearly corresponds to the CHOP INTEND. The CHOP INTEND is a multifunctional test that shows loss of function very rapidly. And so we have very rapid loss of neurons, very rapid loss of function, and then we see a plateau for the remainder of the 2 years before there is infant death. This has been documented in multiple studies. Another elegant study was one done by Kathy Swoboda, and here we see very similar loss of function very rapidly related to loss of motor unit. And this was done in a slightly different way. She correlated both compound muscle action potentials, but actually looked at the number of motor units. This is Mooney, and Mooney is an estimate of the number of motor units. So we have very good evidence that in SMA type 1, there's a rapid loss of neurons very quickly after birth with a plateau in function. And this really, I want to emphasize, establishes a clear rationale for our clinical trial that I'll come back to again. As far as alternative treatments for the disease, we are really at a standstill. There have been small molecules that are found to induce SMN in tissue culture, but when clinical trials have been explored, one with valproic acid with Dr. Kissel, who's here in the audience if you want to ask any questions, it had no clinical effect. Another is phenylbutyrate, which had no clinical benefit. So this particular strategy was unsuccessful. There have also been small molecules that have been identified with high throughput screening, and at the present time, none of these have been tested in clinical trials, and only animal studies have been done. So we really don't know whether these will have a clinical effect or not. The final alternative strategy has been dependent on using an antisense oligonucleotide to alter the splicing of SMN2 and restore a full-length message, just as I showed you, could happen under normal biologic conditions. And this AON requires intrathecal delivery, and trials are currently underway, but we don't know the outcome of these trials presently. So at the present time, we don't have any alternative treatments to what we're proposing here. Now let me talk about the preclinical data, which is, I think, very robust. First, let me comment on the so-called Delta 7 SMA mouse model. This was developed by Arthur Burgess, who's sitting here as one of our members of our team, and this has become the standard model for the disease. And when the Delta 7, the Delta 7 model shows a very near phenotype to the SMA clinical disease, in that the pups are normal at birth. They have a rapid onset of progression with death at 13 to 16 days. And again, as in the patient disease, the CMAP potential declines very rapidly over this period of time. So this has become the standard model for SMA1 type clinical trials. Now, Brian Kaspar and Kevin Faust have done some very elegant studies showing that with a self-complementary AAV9 using a chicken beta receptor and the SMN gene, they can extend survival in this mouse that usually lives 13 to 16 days beyond a year of age if they give if they deliver the gene intravascular on the first or second day of life of the mouse. And the dose that is the most proficient in rescuing the mouse is 3.3 times 10 to the 14 vector genomes per kilo. And you'll see as we move forward that this is our target dose for clinical trial. Here you see here that with the delivery of this cassette in AAV9, we have extension of life really beyond a year of age. And here's the normal mouse life span at 13 to 16 days. Just a couple of slides to show you what the mouse model shows after gene delivery. It's very specific with this dose to the motor neuron pool as identified by GFP and a choline acetyltransferase. Here you can see very nicely the delivery of AAV9 to this pool using GFP as the marker for the motor neuron in this case. And the important, I want to emphasize again when we present the clinical trial that this is a time-dependent transduction. If the mouse is treated at 1 to 2 days, we get 70% of these motor neurons targeted. And by 10 days, doing the same exact experiment, we get virtually no targeting of motor neurons in the mouse. This again emphasizes that the therapeutic benefit is both dose and time dependent. And if you look down here, you'll see that a P1 high dose shows prolonged treatment. As you see here, this is the high dose treatment, this is the high dose curve. We get very good prolonged life in the mouse model. If you delay this, or if you reduce the dose by 5-fold, you significantly reduce the survival of the SMN delta 7 mouse or SMA delta 7 mouse to 35 days. So vastly different from the early high dose at day 1 and 2. And furthermore, if you deliver the dose at day 5, you result in 28-day survival. So again, illustrating that the effects are time and dose dependent. And what's very dramatic here, again, as I mentioned on the last slide, if this treatment is delayed until day 10, there is no treatment effect. And again, this has greatly influenced our clinical trial plan. Now, we have also done studies in non-human primates and using this high dose to illustrate that we can actually target the motor neurons at a time when we would consider doing the clinical trial between 3 and 6 months of age. If we deliver to non-human primate with the same vector with AAV9 at a dose of 1 to 3 to the 14th vector genomes per kilo, we can show very robust transduction within if we treat the monkeys at 90 days. More than 70% of motor neurons are transduced. This is similar to what I showed you in the mouse with GFP marking motor neurons, choline acetylase, choline acetyltransferase marking motor neurons, and then dual labeling, showing that we have clear identification of motor neurons that have been transduced with this dose and with AAV9 as the delivery tool. So the message from our preclinical work is that the treatment effect has its greatest impact with early gene replacement. And early introduction of SMN is a biologic requirement to basically rescue the Delta 7 SMA mouse model. This has also been done in Arthur Burgess's lab, who is here and would be willing to address further questions. He has an inducible SMN mouse model, and this is also a time-dependent induction. If it's very early, it rescues the mouse. If it's very late, it will not rescue. So the message here is clear that we have efficacy that is limited by the dosing range. When we dropped it from 3.3 to the 14th to 6.70 to the 13th per vector genomes per kilo, we had a five-fold less effect. And so this has to be considered in the clinical trial, and also, of course, the time of delivery has to be considered. Now, we had a pre-IND meeting with the FDA and mapped out our strategy for a tox biodistribution study, and this is basically what we came away with from the FDA. We would use the maximum tolerated dose at 3.3, or the maximum effective dose at 3.3 times 10 to the 14th vector genomes per kilo, and we would use the dose at the other dose, the minimally effective dose, at 6.7, and mice would be studied over a 6-month period using the same delivery tool, AAV9, and the same cassette with the chicken beta promoter and the SMN gene. And at each one of these time points, we would look at these outcome measures up to the 6-month time point. So this is the planned biodistribution tox study, which we have now begun. Now, along the way, we've also done non-GLP tox to satisfy our own requirements to be sure that we have a safe delivery vector. And we've done as many as 14 mice treated with the same mode of treatment at high dose, using self-complementary AAV9, chicken beta receptor, SMN gene, delivered at day 1, and mice sacrificed at 24 weeks. And in these mice, we've seen no toxicity. We've also extended the study in non-human primates. This was not a requirement of the FDA, but we felt much better knowing we established clearly that we could deliver the gene to the motor neuron, and we felt much better knowing that we could achieve no toxicity at day, delivering this at day 90, much as we think about doing the clinical trial, and I'll come back to that and explain that further. But we've looked at these monkeys now and sacrificed them at 36 weeks. And basically, we've had very good outcome from this. We've looked at hematology and clinical chemistry values in these non-human primates, and we've looked at organ system histology that was normal. We didn't have any significant antibody titers generated to our transgene. These are binding antibodies, and we did not see any significant T cell response to either virus or the SMN transgene. So these were very encouraging studies that we'll take forward when we present the IND, but these are not our official tox studies. Now, in terms of taking this to clinical trial, we know that we have a time-dependent issue, we know that we have a dose-dependent issue, and we've taken all those things into consideration. And I want to re-emphasize the difference between type 1 and types 2 and 3. Type 1 has 2 copies of SMN. Types 2 and 3 have more copies of SMN and have a more stable clinical course. They don't show this rapid loss and rapid progression of clinical findings. So we think that it is much much more reasonable to try to treat this disease at a very early age, to try to rescue motor neurons before they're significantly lost, so we have some chance of converting basically a type 1 either to a type 2 or 3, or maybe even better. But if we treat the type 2 or 3 patients that are on a plateau, we think that the risk-benefit ratio is clearly less here for the types 2 and 3 than it is for type 1. And again, so these are our issues when putting together the clinical trial. This will be a single-site study, a phase 1 gene transfer trial in SMA type 1. We plan to enroll 9 patients that are clinically affected. They will, none of the patients will have more than 2 copies of SMN2. They will have an age of onset of 6 months or younger, as I've shown you. Most of these kids with SMA type 1 have onset as early as 3 months, and many of them even earlier, at an earlier stage. And so we would be willing to treat them anytime between 0 and 6 months. The exclusion criteria are the major ones. I don't have all of them written down here, but we've presented them in our RAC package. We will not include patients who are currently on ventilatory support or require pulse oximetry, or have a pulse oximetry less than 95% saturation. Obviously, they will not have any other organ system disease or any active viral infection. No immunosuppressive therapy will be used for this clinical trial, and they cannot have AAV antibody titer greater than 1 to 1600 using an ELISA assay. Now, you might ask where that figure came from. We've had experience now in 2 previous gene therapy trials where the AAV antibody titer of 1 to 1600 predicted a lack of transduction, and titers below that were satisfactory in terms of viral delivery and reasonable transduction and good results. So this is a cutoff, but in some ways it's academic because these infants at 0 to 6 months of age are very unlikely to have any significant AAV9 antibody titers. This is a picture of the cassette that we'll be using, and the promoter and CMV enhancer. It'll have an intron. This is the human SMN gene, the same one that we've delivered in mice and monkey, and obviously a poly-A tail. This is a self-complementary vector. It'll be delivered through a venous catheter that will be placed in the infant in the intensive care unit at Nationwide Children's Hospital, and the virus delivered in that fashion. It's this dose escalation study. There'll be 3 cohorts, each one of 3 patients. This is the lowest effective dose essentially that we found, and the 6.7 times 10 to the 13th vector genomes per kilo, the one I showed you that had a 5-fold less effect